the red blood cell. It causes it to be sickled. So right over here I have A, B, C, D. Point mutation in hemoglobin changes amino acid sequence. That causes hemoglobin, I abbreviated it HB because I got tired already, hemoglobin to stick together. Hemoglobin cannot bind O2 efficiently. C, cell shape is sickled. D, cells aggregate and cause vessel blockages. What kind of phenotype am I describing in A? Is that molecular or cellular? Molecular. It's molecular point mutation. Point mutation in the gene, right? DNA molecule, protein molecule. I'm talking about a change in the molecule. Hemoglobin is sticking together. That's B. Is that cellular or molecular? Still molecular. We're talking hemoglobin. C. The cell shape is sickled. Boom! Music to my ears. D. Cells are aggregating. Cellular. Yeah. Okay. Does anyone have questions on that before I move on? Cool. All right. Yeah. What does D say again? D. That's not perfect penmanship. Yeah. Cells aggregate and cause blockages. So the cells kind of, when they have this sickle shape, they'll kind of form these blockages in the vessels. Make it hard for blood to flow through the vessels freely. All right. Okay, so today is a bit more review. So we're going to talk a little bit more about how genes can get disrupted. I'm going to start out by talking about mutations. Let's talk a little bit about transposons today and how genes get transmitted. And then we're going to move on to a little bit of background about how you go from a gene to protein. And that is just to make sure everyone's on the same page before we move forward in this class and start talking about all kind of the molecular details of how these things are regulated. Right? Because I know you guys thought this stuff in genetics, but I don't know how long ago genetics was for you. I don't know if way back then you had the C's get degrees philosophy. If you did, you're going to have time in this class. So change that philosophy. All right. So background on DNA mutations. Mutation rates are really low, it turns out, but are a really important part of evolutionary change, right? So last time we ended with talking about how gene families are made. And one of the review questions was, well, if you have gene duplication, so now you have two copies of a gene, and we talked about how that means, cool, now we have a backup, so at least one of those genes can kind of start to acquire changes without killing the cell. But what happens if no mutation happens? If you just have two copies of the same gene, is that a gene family? No, why not? It's just a duplicated gene. You just have two copies of the same gene. Because if they have the same exact sequence, what's going on at the level of function? It's the same. You can't get functional change unless you change the structure of the protein. The way you get a protein structure to change is by changing the sequence of the gene. Okay, so the most common source of DNA mutation is actually error during replication. Right? So DNA in our cells, we have 3 billion base pairs. That's a lot of DNA to replicate, right? It happens relatively quickly, so mistakes happen. That's the most common form, or the common way that you get mutation. It's just an error. It's a mistake. I had my first biology teacher told me, or professor in college said that the cell is the most efficient machine on the planet, but there's no such thing as 100% efficiency, even our amazing cells. So environmental damage can also cause DNA mutation, but it's usually not direct. What I mean by that is usually it causes some kind of structural change or problem in the DNA, and then the repair mechanism isn't 100% efficient, or it messes up the DNA polymerase when it's trying to replicate it. So a really good example of that is UV irradiation. Does anyone know what UV irradiation does to our DNA, what it actually causes?
Thymine dimers?
Yeah, thymine dimers, you got it. It causes thymines that are next to each other in the strand to inappropriately form these excess kind of bonds with one another. And this thymine dimer, it can't be read by polymerase, so it causes a problem. And so we have repair mechanisms that will recognize these thymine dimers, try to excise them, and replace them with the appropriate nucleotides. But again, there's no such thing as 100% efficiency. Sometimes mistakes are made. Wear your sunscreen. Yeah, question?
More of a general question.
Yeah.
For number one, when you said mutation rates are extremely low,
Yeah,
is that more like in a natural type setting, or like
yeah.
compared to someone who's like exposed to carcinogens or something?
Yeah, you got it, in a natural setting. So if you are like hanging out in a tanning bed, if you start your day by hanging out in a tanning bed or tanning every day while you're chain smoking, you're gonna have higher mutation rates. Every day in the summer, I put sunscreen on my kids before they go to preschool, and oh my gosh, it's like I'm torturing them, and every day I'm like, I'm saving your life. Okay. So a refresher on what types of mutations exist. So this is terminology, right, that I'm just going to expect you to know for the rest of your life, and especially in this class. So here in this chart, it's kind of walking you through the first three mutations. So here we have the codon that exists at the level of DNA, TTC, and encodes a lysine. So silent mutation, so the third position of any codon, does anyone know what that's called? The wobble, right, okay, go you guys. So the wobble, right, you can have variation there, and oftentimes it won't, sometimes it does, but oftentimes it won't change the amino acid that's encoded. So that type of change is referred to as silent, because even though the nucleotide sequence is changing, it's not changing the protein, and that's really at the end of the day what matters, right? If it's encoding a protein, there's a mutation, but it doesn't cause a structural change in the protein, nothing's gonna happen. A nonsense mutation is the type of mutation that often causes the most kind of deleterious effects in the protein, because nonsense means that the nucleotide change, in this case, switches from a lysine to a stop codon. Right, so if you have a stop codon that's all of a sudden exists in the middle of a protein, a lot of times the protein, it can't even come close to folding appropriately, and that can either cause protein aggregation in the cell, or the protein usually just gets degraded. So it doesn't even have a chance to function. So nonsense mutations almost always cause non-functional proteins, right? If it was at like the second to last codon, maybe not. So it depends on the context, like where is it happening. But by and large, these are regarded as the most damaging types of mutations. Missense mutation changes the amino acid, but there are two subcategories of missense, conservative or non-conservative. Right, and this comes down to what's the amino acid, how similar or different is it from the original amino acid? So lysine, right, that's a positively charged amino acid. If it gets changed to arginine, it's structurally pretty similar and also positively charged. So that might not have a really great impact on the structure of the protein. Whereas if it gets changed to threonine, that has totally different biochemical properties, right? That has, it's structurally different and it's negatively charged. So yeah, you can bet that going from a positively charged amino acid to a negatively charged is going to affect the way that protein folds. Yeah. And then finally, the one that's not shown on this chart